I'm going to talk today about fully mining RNA-Seq data. I'm going to stop. I'm going to start right there. Put me on this. Right here. Just like, no, I'm not going to do that. All right. So I'll talk about fully mining RNA-Seq data. Let's see. All right. So to understand my talk, and I'm sure 90% of you are very comfortable with what a microRNA is, but if you don't know, you won't understand my talk. So just very briefly, what are microRNAs? 20 nucleotide non-coding RNAs. This is my bread and butter. This is what I started off on. Again, my doctoral thesis was starting with microRNAs. There's about 2,500 of them annotated now. What's more striking to me is that in the last decade, there have been over 40,000 publications on microRNAs. I was fortunate enough to be there at the beginning, but what do? They regulate gene expression by base pairing with complementary sequences and target RNAs. The partial complement there is message degradation. A complete complements message degradation, which is great for transcriptomic studies. Partial complements are more difficult. That's where you're looking at translational repression. All right, what's the big picture with microRNAs? This is, again, where I became very interested in them. It's about 2001, first papers came out. I know there was one in 1993. In 2001, the first papers came out in humans. They showed, hey, there's about 50 of these things, and they're regulating genes. At the time, I'm working in neurodifferentiation, and I've got this huge poster board, and I've written down every single interacting player, neurodifferentiation, notch, delta, wince. I'm like, what the hell am I going to study, right? And at that time, these first papers came out, and all of a sudden, you've got a short RNA that's capable of coordinating the actions of 20 or 30 different genes. So boom, I'm sold. I spent a year fighting with my PhD advisor, trying to get a project started on this, and then ultimately I did. And now I work mostly with microRNA misexpressions and how they contribute to oncogenesis. All right, so I work with microRNAs. That's the start of this thing. The other part that you should be aware of, a lot of what I'm going to talk today, the data set that I'm going to talk about, is a comparison between two different breast cancer cell lines, MCF7 and MDA-MB231. Right? MCF7s, now the thing about this I should start with is they come from fairly similar locales, right? So all intents and purposes, you would think they would be fairly similar pathology, they're very similar oncology, but they're not, right? MCF7s, they do not migrate, they're not very invasive, they respond very well to chemotherapy, they're very hormone responsive, right? So MCF7s are very docile, whereas MDA-MB231s are much more aggressive. They're much more metastatic, much more invasive. They're unresponsive to hormones. They're resistant to chemotherapy. Why did I study these two? Well, they're the two most commonly studied breast cancer cell lines. So I believe you're probably familiar with that. The other thing is that there wasn't a transcriptomic study. Everybody's done microarrays comparing MCF7s and MDA-MB231s. But as of a year or so ago when I did this analysis, nobody had reported a comparison between the two from a next-generation sequencing standpoint. All right, so my question initially was what are the relevant microRNA differences in the transcriptomes of these breast cancer cells? All right, so what did we do? I did a MCF7 MB231 RNA-Seqs, did two different ones. I did size-selected, sequenced the small RNA populations, sequenced the total RNA populations. I'm very interested in hearing about the longer reads. So I've got the two data sets. One, the small RNAs, I can identify what small RNAs are present, and then I can also look at the mRNA levels. All right, total RNA-Seq reads, just to give you what the data set that we're working with here. I got about 50 million reads for MCF7. This is total RNA or longer RNA-Seq. MDA-MB231s, this is Illumina. I got about 75 million high-quality reads, good Phred scores, right? MCF7s, I know I got the right stuff. MCF7s express the estrogen and progesterone receptors. MDA-MB231s do not. That's what they should say. Right, small RNA-Seqs, I got a little over 20 million reads for each, high quality. Right? Now, I think for a lot of people, I've got, in the next couple of slides, I'll show you, I did what I set out to do. Right? I found there's very little difference in over 2,500 microRNAs, 2,549. There's essentially no statistical difference between them. And then I found 39 that were significantly differentially expressed between the two cell lines. Right? So those 39, what was comforting, I would say, is that those 39 are already known to contribute to breast cancer pathology. All right? So on the one side, it's like, well, you found 39 differentially expressed microRNAs, but they're already known to contribute to breast cancer pathology. Did you really find anything? And I would say yes. I got very excited because I actually identified 45 differentially expressed microRNAs between MCF7 and MB231. one, and 6 of them had no known involvement in breast cancer. That's what I need, right? I've now looked at all microRNAs in the 2 different cell lines. There's a subset of them that are differentially expressed. They've already been reported. I've got 6 that have. This was the groundwork for a foundation award I got earlier this year, right? It's great. So now I'm going to study the effects of manipulating these 6 particular microRNAs in MCF7s and MDAs. You know, will they make the MCF7s more aggressive? Will they make the MDAs less aggressive? Right? Now, what else did I have? I also did the poly-A-selected total RNAs. Right? So now I've got the microRNAs that I'm interested in looking at. I've also got the long RNA reads. Right? So I've got the long RNA reads. I can identify which mRNAs are present and potentially regulated. I also do a lot on microRNA target prediction. Here's one of my recent papers looking at that. But this is where things started getting interesting. It was already interesting. I already got money and all that stuff. But this is where it got interesting to me. An initial examination of the potential target sites. So microRNAs regulate mRNAs by complementary base pairing, right? Well, when I started looking at complementary base pairing and I started looking at predicted targets for these microRNAs, what I find is that several of my predicted targets had been disrupted. What do I mean they'd been disrupted? They weren't what they were supposed to be according to the genome, right? It wasn't SNPs. I'll talk about that in a minute. It wasn't error. There's a high quality reads, right? So what is it? Well, it turns out it was RNA editing. Right? What I mean is RNA editing. What is RNA editing? The main one that you're going to see in transcriptomic data is A to I, right? It's catalyzed by an enzyme called ADAR. It changes adenosine. Never use a pointer. I like the mouse. This is a beautiful built-in pointer. All right, it changes an adenosine to an inosine, which is essentially converting an A to a G, right? So when I'm reading through my transcriptomic data, if I see an A that now looks like a G, I'm pretty sure that that's an A to I edit. I'll talk about that. I do a workshop about how to mine or look for potential edits, and that's something I'll talk about how to be sure. But essentially, there's been tens of thousands of these things identified recently. Most of those are in 3' UTR sequence. I'll show you what we found. Now, I see a bunch of my target sites are disrupted. Now, is that relevant to what I'm doing? It turns out it is. There's been a lot of reports that you have global changes in editing in different pathologies, right? And for us, it was particularly interesting. Increased expression in editing by ADAR is fairly characteristic of breast cancer. All right. So now I've got some microRNAs, and it looks like, as I'm saying, it looks like it's disrupting my target site. So how does RNA editing affect microRNAs or relate to microRNAs? Well, there's already been some reports showing that they are involved with one another. If there have been reports showing that Drosh and Dicer processing is altered by microRNAs, they can go into the hairpins that microRNAs are processed out of. That you can put edits there and actually change the species of small RNA that gets processed out, or which side of the hairpin gets processed out. So there is a relationship there. There's also a relationship. There was a paper, I should have put the citations, I don't remember. Japanese group showed that changes to the microRNA seed region, this is the part of the microRNA that is necessary, not necessarily sufficient, but necessary for microRNA binding, that changes in the seed region can change what the mRNA targets are, right? But what we were seeing and what we were suspected, or the question became, can ADAR editing of 3' UTRs actually affect the microRNAs that can bind to it? All right, so that's a new hypothesis that's developed out of this work. Right, initially, I was looking at which microRNAs are differentially expressed between the 2 cell lines, what they can do. But now I was very intrigued by the fact that I thought ADAR editing of mRNAs could actually alter the regulation by microRNAs. All right, so a second project evolved out of this work. All right, so what do we have? We have next generation sequenced the poly-A selected RNAs from both, right? And then we're going to identify their edit sites. And then I'm going to look at those edit sites for their potential to alter microRNA regulations. Here's an example of one. I've got a bunch of reads over here on the left. Well, I guess left and right. And what you can see is if I line them all up with bow tie or top hat, right, do a cluster W, you can see that in most of the reads you've got a T at this position. This is negative strand, so you have to, it's A to G instead of T to C. But so here's a bunch of T's, should be A's, have been converted to C's, should be G's, right? So a percentage, but 20% of my reads, I've got G's where there should be A's, right? So this is fairly characteristic of what you see with RNA editing. It's usually not the entire population. I'll talk about that too. All right, so we did that. I went through and I predicted all of the edit sites in the total A polyselected, and then I examined them for their potential to alter microRNA regulations. How do I do that? I take that seed region of a microRNA, right? I just compare across unedited sites versus edited sites. I look, see if you can see over here on the left, if it's in an unedited, I could potentially have a microRNA that doesn't bind, that if it was edited, it would bind, all right? So I do that. When we looked across the full spectrum of microRNAs, we found 96 microRNAs. Not most microRNAs, this has no effect on whatsoever, right? But we find a subset, about 96 human microRNAs. There is a significant alteration in their complementarity to mRNAs based on editing events, right? Here's a good example. In edited, if we took all of our edited sites and edited them, we had 258 complementarities to seed matches to miR513 with 0 complementarities to our potential edit sites if they were unedited, right? So here's another, miR140. This is one I'm particularly interested in. miR140, and the unedited sites, I had 0. This is flanking sequence. I'll talk about this in later talks. I see roughly really no enrichment for miR140 complementarities in these edit sites or around these edit sites. But in the edited state, I've got about 80 targets for miR140, right? So now, is this relevant to my work? And yeah, it turns out that miR140, with no real rationale as to why, several people have reported that miR140 is involved with breast cancer pathologies. All right, so now things interest me. It's very interesting to me at this point. So now I've got a set of edits that we've predicted in our data. I've got a set of complementarities, right, to those edits. Now, the thing that I find very striking is when we looked and we separated it out, and I looked at MDA MB231s, I found that this one target that we focused in on, DFF alpha. DFF alpha is an apoptotic regulator. I know that miR140 is related to cancer, et cetera, et cetera. There's a bunch of rationale, but I was really interested in DFF alpha. That got really exciting when I looked at MBA MB231 edit sites and found that none of them exhibited editing, whereas on MCF7s, the majority of the mRNAs were edited at this position that created a complementarity to miR140, right? So now what do I have? I've got DFF alpha. It's edited in MCF7s. It's not edited in MDA MB231s, right? So what would that predict? That would predict that it could be regulated in DFF alpha or in MCF7 and not in MDA MB231s. And I'll show you some data to support that later on. All right. So I started off, I just identified complementarities or differentially expressed microRNAs. I found a subset that were undescribed. Out of that work and looking at targets, now all of a sudden we're seeing that RNA editing is playing a role in likely regulation of mRNAs, right? So what does that mean for this work? Well, miR140, I'll talk about that more later on. Single base substitutions caused by ADI editing can have dramatic effects on microRNA targeting. For this group, maybe the most important point is that it's necessary to look at transcriptomic data to make accurate microRNA target predictions. One of the huge mistakes I feel the microRNA field makes is that they use genomic sequence for their target predictions. Well, if some of the targets only reside in transcriptomic data, you're never going to figure out what those targets are, right? I think that's a major obstacle, one of the things that's impeding accurate microRNA target prediction. All right, so that's awesome. I've got that. We're in the process of putting that work together. Presented it in Keystone a couple months ago. Our manuscript's about ready on that. So yeah, we're done. We found 2 really cool things out of this transcriptomic data, and then I got this graduate student who comes up to me and he's like, hey, wait, let me show you something. What do you want to show me? So whenever he was looking at the differentially expressed microRNAs, what popped out at him was that he also saw, in looking at very similar sequences, that there were 13 SNO RNAs significantly overexpressed in MBA MB231s than MCF7. It was weird. It was only the MBA MB231s that you had this overexpression of SNOs. Well, that's great, right? Your gut reaction is, who cares? It's a SNO RNA, right? SNO RNAs they alter, edit ribosomal RNAs, tRNAs, right? Who cares? Well, no, who cares? But the truth is, there are 13 of them. See, this is why I don't use these things. They don't work. Oh, there's 2 buttons. There's 2 buttons. Okay. So I've got 13 SNOs that are reproducibly overexpressed and pretty significantly. 27, 18-fold more expressed in MDA MB231s than MCF7s. Again, what are they? SNO RNAs. They guide chemical modification. I'm actually using the pointer. I never do that. They actually do the guide chemical modifications to ribosomal RNAs, tRNAs. Now that said, about a third of SNO RNAs are called orphans, right? They're orphans because we don't know what they're editing. We assume they all do the same thing, right? So then he shows me this. I call him Pickle. Dylan, Dill. I call him Pickle. So Pickle comes up. He's going to Emory, right? It doesn't matter. All right, so he comes up and he says, hey, look at this paper. And what a couple groups have now shown that some SNO RNAs are SNO RNAs. Most microRNAs are microRNAs. There are some SNOs that are behaving like micros, right? What does that mean? So some SNO RNAs will go modifying ribosomal RNAs, and then a chunk of them will get cut out and work like a microRNA, right? Some of them, most of it goes to microRNA, but you still get some editing, some of it, maybe occasionally get a piece cut out, but it seems like there's a blur between SNOs and micros. Again, for me, this is super exciting. I'm a microRNA guy. You can tell me that there's more microRNAs than we know about. I'm on board, right? Now you also say that we can show that those things are overexpressed in my cancer model. I'm definitely on board. All right, so we introduce a term, sdRNAs. This is SNO-derived RNAs. It's like a microRNA, or sdRNAs. So are our SNO RNAs being processed, right, so we know that there's these 13 SNOs are being edited, or they are being processed into microRNA like RNAs called sdRNAs, right? So what do? So now we're going to look at processing. So we take our small RNA transcriptomic data, right? On the one hand, we can use it to see the larger molecule, the 200 base pair molecule, right? But then we can also look for the short sequence root. And what we found is that all 13 SNOs that were overexpressed in MDA-MB231 were being processed into microRNA-like sdRNAs, right? Super exciting. Now, not only are they being processed, they're being preferentially processed in MDA-MB231s in the RNA-MCF7. Right? I get about six. So there's only about, I'm looking at SNO93. SNO93 is overexpressed about 27-fold in MDA-MB231s. If I look at the small fragments are overexpressed about 75 times in MDA-MB231s as compared to MCF7. Right? So they're not only are they being overexpressed, but they're being selected for processing preferentially in MDA-MB231s. Right? So now I'm excited. I get excited easily. I don't know if that's coming out. All right. So now what's more, we do some. I'm gonna go back to the literature, guys. We go back to the literature, and what we found is there's a group that's already showed that SNO93. They use a different name, so we changed it a little bit. SNO93 is being processed into an sdRNA, and that can function in a luciferase assay. They don't show a functional relevance. They don't show what it's doing physiologically, right? But they show, hey, it is getting processed. And once it gets processed, it can function like a microRNA, right? So, and we looked at it, they found the exact same piece that we find. All right, does that overexpression of SNO93 sdRNA contribute to the pathological differences between our two breast cancer cell lines? That's the real question. Here's the piece. Now we find two pieces that are coming out of the SNO, base pair 2 through 20 or 3 through 30, but that's it. It's not like the SNO RNA is being cut at random. Right, it's always this left-hand side of this hairpin, right? Sometimes it goes to 20, sometimes it goes to 30, right? But it's always this left-hand side, and those account for 98% of all the fragments of SNO193 we've ever found. What's in green right there? So it's a specific location that's being excised, right? So what do we do? We made antagomeres to that. We threw it into what was this? We throw this into MDA-MB231s, and what we find is it come out about 48 hours. It didn't matter if we used inhibitor 1 or inhibitor 2. That's to the left green or the right green. Whichever inhibitor we used, didn't matter. At 48 hours, we had about a 20% reduction in cellular proliferation of MDA-MB231s. These are the hyper-aggressive, invasive cell lines, right? So we can slow their proliferation by about 20% by knocking down this sdRNA. Okay, that's cool. What else? Not only does it inhibit proliferation, but if we inhibit that small fragment, we also knock down invasion. If you're looking at an invasion assay here, you take your cell lines, you plate them on a media, you let them invade into the matrix based on some signs. After they've invaded, you give them a certain amount of time, you wipe away what's on the surface, right? And then you just count how many have invaded, and what you should be able to see fairly by eye, it's not, you've got more over here on the left in the control. If you quantify that, we've got about over 40% reduction in invasion when we knock out that sdRNA, those sdRNA fragments. In our MDA-MB231s, we see nothing in MCF7s, no changes, right? So, okay, so there's something there. This is also work. This was Pickle's thesis. We're in the process of putting that together now. All right, or as a manuscript. So what are our conclusions from this work? SNOs are often processed into microRNA-like sdRNAs. SNO93 silencing reduces cellular proliferation and invasion. Does not affect cell migration. I guess that's a thing. The work. This is the first ever characterization of a functional role for an sdRNA. We're very excited about this. And SNO RNAs, and likely other types of noncoding RNAs, their expressions are also relevant to transcriptomic features, right? And they could directly contribute to cellular phenotypes and pathologies. We're not just talking microRNAs anymore. We're not just talking mRNAs. There's lots more there. What else is there we haven't even looked at yet? Right, well, we haven't looked at mRNA profiles. I've done a little bit just to see if the right things are there, but I've done very little with mRNA profiles. Right, we're way beyond microarrays. But you know, it's nice. We can look at start-stop sites, splicing variants, etc. I can do a more comprehensive editing comparison. I plan to do that here shortly. Another thing that we're going to look at is additional small non-coding RNAs that are differentially expressed. scRNAs, tRNAs, snRNAs, small RNAs corresponding to repetitive elements. Very intriguing. Right? The big one that I want to get into, which is why I'm very interested in Dr. McComb's talk, or McCombie, excuse me. We're going to screen differentially expressed long non-coding RNAs. Very exciting work. The guy got me at a Keystone conference a couple months ago. He's like, well, why are you working on microRNAs? Why are you working on mRNAs? It's like from being from Botswana or being from I don't know, Holland, right? When long non-coding RNAs represent China, the population, the body of long non-coding RNAs in the transcriptome is so massive, and we're not even looking at it at this point. And I haven't looked at my own data, and that's kind of my next goal. So again, the title of my talk should have been fully diving into, fully mining next generation data sets. I feel like I could have stopped after I found my 6 microRNAs that had been differentially expressed, right? But I just keep finding new revelations from this one series of experience. Now that said, I'm guilty too. I've got about 10 projects running right now that I need to be diving into, but I'm going to stop. I'm not sequencing anymore. I'm done. I'm going to go with what I got and figure out what's there, and then we'll proceed. Now, I've been very fortunate. I got to know at my lab. I've only had my lab for about 3 years, so we're rolling, right? I've been very fortunate. I've got a career award now, an R01, a foundation award from my university. Yeah, everything's going very well. And I would be happy to take any questions.
Questions, please. He's definitely rolling. I can tell.
Thanks, Dr. Borchert. That's a very nice presentation.
Thank you.
I think you raised two creative questions I have thought about. The first, you see the RNA edit between the target and the microRNA. I think the coevolution between target and microRNA, this kind of thing should be the events for coevolution. You think which way is drive force, which way are consequence, or hard to talk
I think
about?
they evolve. You're 100% right. They evolve together as a common regulatory system. Starts with I've published a couple of reviews actually on microRNAs and their targets, right, coming from the same repetitive elements. Now I can show you, I can conclusively demonstrate to you that at least 50% of microRNAs were originally formed from transposable elements, right? So what I see is you've got a transposable element jumping around the genome, right? And at some point it creates a microRNA by inserting in one strand, inserting in a neighboring position in the opposite strand, making a hairpin, right? Now in the time it takes that to happen, it's also jumping into 3' UTRs, right? So you get coevolution, the generation of a microRNA and the generation of targets are happening simultaneously as you colonize a genome. Right now, I talked to a group from IBM, and they went through the genome and they found about 99,000 potential microRNAs. Clearly, we don't have 99,000 microRNAs. But so you get the creation of these things. If it has a positive, what's the words? if there's some benefit associated with the maintenance of that regulatory system, then it'll be maintained. Otherwise, it's lost. So these things are being created in unison all the time and then fading away. So I'm 100% with you. I believe microRNAs and targets come together. Right? They don't arise independently; they arise together. So yeah.
As an analogy for the virus.
Yes.
In the smaller elements, genetic elements, to be mutated, then follow up by the targets, is that possible? Or whatever is pretty equivalent for the target and evolution of target and in terms of the
RNA editing. So RNA editing, I think, was originally a system to control viruses and control transposable elements. So the editing is still you're still gonna, I think you're gonna have a higher percentage of editing in targets and a higher percentage of editing in microRNAs compared to the rest of everything else because they do have a viral or transposable element origin.
I so many non-coding RNA, microRNA, small RNA, tRNA, derived RNA, non-coding RNA. But you think there's kind of so complex spaces for regulatory RNA. So in what kind of situation, what kind of tissue, which spaces is more focused, for example, in the tumor environment? So which spaces is more,
Is the
you know,
whatever's differentially expressed.
critical for our to focus on? Yeah, thank you.
You take a system, whatever is the most differentially expressed. That's what I like. I don't like stuff that's off a little. I like this is off 75-fold. Right? That's what I would say. I think in different situations, there's different things, but it's always going to be a non-coding RNA. That's my. I know everybody's like cancer is because of P53 and ATM. No, most of the time.
Yes, we do all choose one based on your. Okay, thank you.
That's one thing we got to do is get to the point where we comprehensively look at everything, not just mRNAs and et cetera. Right?
Okay, more questions?
Thank you. I sort of came in a bit late, so this may be out of context, but have you come across the reverse phenomenon where there's an editing of a CG back into an AAT? And what impact does that have,
I
if
think
any?
that does occur. And the other thing you got to take into account is this A-to-I editing is one of 200 edits that we're aware of. Right? We think it's the most prevalent. We think it's the most prevalent because it's the one we can see looking at transcriptomic data. Right? An A turned into a G. All right, I get that. There are 200 different RNA edits. And are they C to Ts? Yeah, I think those are going to be relevant. And I think it's going to be a lot more relevant. But honestly, we don't have the technology right now to even look at those things. There was a group in Japan that just came out with ICE, and that's it's a different way to sequence A to I's where you actually see them. I mean, again, we're not looking at a fraction of what edits are actually there, and they're all going to be relevant.
Hi, I came a little late, so again, I might have missed this. Have you looked at these snow RNAs and the conditions of stress or hypoxia with arginine treatment or something of the sort?
No, not specifically. I do some work with hypoxia. I found a lot of mutation in hypoxia, actually. More specifically in regulatory regions. But I think your snow RNAs, I mean, I have to look specifically at that scenario. And I'm not going to because I'm done sequencing. Done. No more sequencing. I got a lot of sequencing to do.
Hello, I have a couple questions just to avoid to race to conclusions with results, since some of us might be working with data ourselves and doing analysis. One of your slides, you showed that there was a bimodal quality distribution. Why was that, and how did you address it? And then two, did you work with the usual nucleotide bias in your reads? I imagine, since you said these are single reads, they're probably short. And did you trim that, and what was your remaining length after you dealt with the nucleotide distribution bias?
So I do have another talk coming up. I'm going to talk specifically about, I have a workshop later. I'll talk specifically about trimming and quality scores and things of that nature.
Okay.
I got a workshop, I think, at 1.
And the other thing, since the end...
Slides and everything about that.
Sure.
Okay.
And since the other thing, too, is with respect to the end goal of identifying RNA editing events, your mapping, most mappers are end-to-end. Did you do local realignment? How did you ensure yourself that your editing events, your candidate editing events, are not false positives with respect to your mapping?
Again, I'll talk about it. We took out all the SNPs, we took out all perfect hits, and I'll talk about that in the workshop.
Okay, just another question would be, so in your targets, you have multiple UTRs. An editing of one UTR event doesn't necessarily mean that you've protected yourself against mRNA degradation of your target. So what was the ratio of number of...
Usually with an edit, you see about 20-30% of the mRNAs are going to be edited, which means 20 or 30% of your mRNAs can now be controlled by the victim microRNA. And it's not an on-off switch. It's not an on-off switch. What we're looking at here is you have 30% less coding, right? Which can have
So,
It's never an on-off. It's always a lowering the levels and increasing.
so if you have multiple mRNA binding sites in UTR, you see perhaps one of those being edited, or
Within a single UTR.
yes.
Actually, I haven't looked at that.
So you see conservation.
I will say what you do typically find is that ADAR, when it locks into a UTR, it hits all of the positions. I haven't looked at it specifically, but when ADAR locks into an mRNA, it tends to hit multiple positions side by side. So
Okay.
you tend to make it right now. I can't say for sure because I haven't looked at that. I know from previous data that I've analyzed, what you'll find is that if you've got a single transcript, you'll find multiple positions within that transcript have an extra.
Okay.
Now, I'm not a big one for multiple target sites in a 3' UTR either. I think you just need one. But that's a good debate. You have to hear and sit down and fight over that one all day. Right? I think you need one good target site.
Right, that's why I was asking the question. If you looked at that number of sites, number edited, and distance down the UTR, what that distribution looks like.
Right, no.
Okay. Thanks.
Okay, any more questions? If not, let's thank Dr. Borchert again.